Okay, so I'll tell you a bit about, I just called it Plants, Worms, and Global Food Security. This kind of sums up the kind of work we do in the lab. Specifically, because global food security is super broad, we're interested in pathology, right? So understanding and trying to mitigate the crop losses imposed by pests and pathogens. So hopefully this is not news to you. You should know, and I hope you know that pests and diseases are a major constraint on global food security. But there's a couple of things I want to point out from this slide. The first one is that the average loss, yield loss from pests and diseases, 20 to 30%, is not the mode. So it's not true that everybody loses 20%. So that would be bad, but it would be totally manageable. But the reality is that some people lose zero and some people lose everything, and it's that sporadic instance of pest and disease that gives rise to the famine and the true food insecurity from pathogens. And the second thing I want to highlight on this slide is that if you ask a farmer what is the worst pathogen for your crop, and then you go and measure what is the worst pathogen from your crop, those lists don't always line up. Okay, so there's something called orphan diseases, so diseases that are objectively important but under appreciated even by the farmers themselves. So it's not going to surprise you that I work on one of these, so plant-parasitic nematodes are one of these orphan diseases. And you're probably thinking, like, how can it be possible that a farmer can't realize that this is the most important thing for their crop? And it sounds stupid, but actually it's not. It's very difficult to attribute the crop losses you see to something you can't see. And so these nematodes are root parasitic, soil-borne, out of sight, and so they get literally and then obviously metaphorically overlooked, both by the farmer but also in some cases by the research community. So this video that's been playing shows you one of these plant-parasitic nematodes infecting a root. So what happens is they burrow inside the root, they get to the middle of the root, and then here they drain the plant resources. They swell up a bit like a tick, and so these dots on the side of the root here are this nematode. And in draining the resources, that's how they contribute to the crop losses due to this disease. So these nematodes are really difficult to work with, and some of the work we do in the lab is kind of fundamental biology on the system, and some of it is to try to make it easier for us to work with. And the thing I'm going to tell you about today is like the most fun experiment I've ever done, for sure, and you'll see why in a minute. and the reason why we got into this in the first place is because we were just trying to make them easier to work with. But in so doing, we did something that turned out to be quite important, and that is being holistic and being dynamic in our understanding of this pathology. I'll explain what I mean. So typically in a pathology, you'll look at like one part of the plant and a snapshot in time, and you'll quantify whether or not there's disease or something like that. And so by being holistic and dynamic, I mean we've started looking at all of the pathogens on the whole plant for the whole time. And this has completely changed our way of thinking about how to develop a solution for this problem in the field, and I'll come back to that at the end. Okay, so one of the major bottlenecks that we set out to lift in our field is phenotyping. So this is just answering really what sounds like trivial questions. So how many nematodes infecting this plant? How many nematodes are you going to get in the next generation? And how well are they doing? This kind of question sounds simple, but it's actually really difficult to measure in practice for the same reason that these are root parasitic, so they're hard to see. So we decided we wanted to just lift this constraint, and we came up with a four-step plan, and our four-step plan looks super intuitive. So we want to have a system where we can see the roots, we've got to be able to see the nematodes, we've got to be able to measure the nematodes, and then we want to scale that capacity to tens of thousands of plants a day. Now this was a big ask, and so we decided to start in the easiest system we could find, which was the model plant, Arabidopsis thaliana, infected with beet cyst nematode Heterodorus acti. This is an example of what this pathosystem looks like. So this is this sort of small white lemon thing that looks like a lemon is the nematode infecting the root. The way the system works is you have this 5-centimeter petri dish, you put a single plant in the middle, it grows down, and then the root system will hit the bottom of the dish and then fan out. So your 3D root network becomes 2D. And then when you infect with nematodes, they will go down and infect your 2D plane, so you end up with a 2D plane of your pathogens. And then what would normally happen is a human would take this, flip it over, look under a microscope, and count the lemons, count the lemon-shaped things. And so previous maximum speed was 3 lines a day, okay, per human, per trained human. So if you want to scale this to thousands of plants a day, you're not going to be able to brute force this. You're not going to be able to convince humans to do this, right? So you need a machine. I think that's clear. Now, there isn't a machine that can do this, right? So you need an idea. And so the idea we had was we would buy a 3D printer, and then we would just design and 3D print a machine that would help us to do this. So, so that's what we did. We went through this really like exciting process of just a couple of months, this October to November, of this kind of rapid hardware development, starting from these very kind of humble beginnings up to slightly less humble endings, where we designed and printed machines that would help us solve this problem. We boiled it down to one engineering problem, which is just moving Petri dishes. So we thought if we can just move Petri dishes, then we can move them under a camera, take an image, phenotype from the image, for example. So we started going through this process, and so in November we had the first machine that looks simple because it is, where you give it 3 things, and it moves them, and it gives you back 3 things, okay? And while this looks simple, if you can do this, you can solve the phenotyping problem. And so then, you know, we're pleased with this, and we scaled it up, so this is now using the full-size Petri dishes. And then by end of November, we added all the other bits, so computer, camera, lights, etc. And so you can see this version will take 1 dish at a time, take an image, and, you know, continuously process the dishes. Okay, so by December, we had scaled this up to the kind of final version, if you like. So this is what we call hardware 2, and you can see how this functions. It takes 1 Petri dish at a time, pushes it into the imaging chamber, gets illuminated, takes the image, and then spits it out the other side of the machine. So this is real-time video. So each one of these machines would do 1 plate per 6 seconds. And so I said to the team, we want to do a plate a second. And so they just printed 5 more machines, got 6 machines, you can just do 1 per second. And so now we can do 10,000 plants in 3 hours. So this is a really nice picture, right? So this is what the images look like when they come out of the machine. And so if you've been looking at nematodes for your whole life, then you will appreciate how much of a nice picture this is. So you're, and if not, take my word for it. So what you're doing when you're looking at this is you're looking up at the root system, and you can see the whole root system, and you can see all the nematodes. And so this is, so the white dots, right? And so this is what I mean about being holistic. You can see all the things on the plant at the same time. Now, obviously, human would still have to, you know, count the nematodes, so we need a machine to do that too. So we collaborated with computer vision and AI team to develop some nematode recognition software. And so from this, you pass it the images, and it does 2 things. It labels them, and then it quantifies them as well. And so you can see on the right-hand side, if you're comparing how many nematodes the computer thought was on this plant versus how many nematodes the human thought was on this plant, then the correlation is really, really good. So the computer is as good as a human at counting nematodes, but computer is better than human, right? So computer can do things that human can't do inherently, like it gets the shape of every nematode, it gets the size and the color and the roundness and the opaqueness, and all these kind of phenotypes that would be effectively impossible to measure if you're a human, and yet are important in agriculture as the biology of the system. So really phenomenal capability. And the final phenotype that's actually the most important is the position of every nematode. Now, this doesn't sound like the most important phenotype, but if you consider that the phenotyping is non-destructive, you can image the same plant tomorrow, and provided you have the position, you can link today's nematode with tomorrow's nematode, and so then you can track the dynamics of those phenotypes over time. And so this is basically what I mean by this holistic and dynamic pathology. So you can track the dynamics of this entire pathology, all the nematodes on the plant for the entire time. So this is just a representation of that. You can see some images from a much larger series over a 90-day period. And what we can do is take these images and then line them up and interpolate them to make a video where you can watch the nematode grow. This is 90 days. And then you can watch the nematode lay eggs, you can watch them hatch, and they will go and infect other parts of the plant. You'll see that over here and over here. And so this is, you know, for someone who was counting lemons on a microscope, right, this is crazy capability. And so we thought this was really cool. And so we decided to deploy this kind of holistic and dynamic pathology in the largest nematode infection trial in combined history of human race. So that's what you can see here. So this is 10,000 plants. So you've got 5,000 on this bench, 5,000 on this bench. So it's 550 different varieties of Arabidopsis in this case, each with 20 biological replicates in a randomized block design, and you can see some of the lab setting them up. We put the 6 machines that we printed into the greenhouse. We put gazebos to protect the humans. This is one of the humans. This is Siwan. PhD student working closely on this project. And then every other day for 90 days, 4 people from the lab went into the greenhouse and imaged all 10,000 plants through the machines and then put them back in the same place. So this amassed like 400,000 phenotyping events and tens of millions of nematodes. So a lot of nematodes, right? To put this in perspective, we know this is true because we measured it. This is more nematodes than have been measured in all previous publications combined and then doubled. So this is a lot of nematodes. And then for every nematode, we know not just like how big is it, what color is it, how round is it, but we also know like what is it going to look like tomorrow or in 2 weeks or yesterday, or what did the root look like before it even got there. Because although it's 400,000 events, it's actually 10,000 movies over the 90-day period, like 45-frame stop-motion movies. So, you know, we thought this was quite cool. And so then what we've been doing is now, with the AI team, extracting phenotypes from these movies so that we can then do biology. And so I'll tell you about 2 of the kind of things that we're doing, just one example for each. The first one is trying to understand some new biology about this system. So what is a new fundamental feature of this pathogen that we didn't know before? I'll tell you about that. And then the other one is to try and use this capability to do something useful. So for the first one, the fundamental thing, so I'll pose a question to you first. So the question is, nematodes can infect all of the root. So if you have 2 nematodes that are infecting the same part of the root, do they help each other or do they hurt each other? Okay, valid question. And you can totally guess it, you know, you could justify it either way. I'm a nematode sympathizer, so I had assumed that they would help each other, but I was wrong. So definitely they don't. And we know this because from 60,000 biological replicates, we can go to every nematode, we can draw a circle around it, and then we can just keep enlarging that circle until we touch another nematode. And then this is like your personal space. Okay, so this is the space until you get to your closest neighbor. And then you can plot that on the x-axis. And then you can say, and how big were you? And plot that on the y-axis. So how well were you doing? How much nutrition were you getting from the plant? And what you can see if your closest neighbor is 1,000 pixels away, so half a plant in this case, or 100 pixels away, you are the same size. It's like a flat line. But if you have a close neighbor really close, then you get this really strong skew to the low end. And so our interpretation of this is that if 2 nematodes are infected in the same part of the root, then they compete with one another for resource from the plant. So, you know, there's other things that we learn, but I just think that, you know, for me at least, to learn something new, and this is a fact, by the way, so because of the, you know, number of biological replicates, the p-value associated with this is like 10 to the minus 170 or something. It's just definitely true. And so it's nice to be able to learn something that is just absolutely, you know, a new fact about a system you've worked on your entire career. So that's the, yeah, kind of fundamental stuff. And then for the more applied stuff, trying to be useful, for this I have to tell you that it wasn't just a random set of 10,000 plants, right? So it was actually a mapping population. And so we actually know the genes of every plant in that 10,000 set. And so what we can do then is we can correlate the phenotype of each one of those plants, i.e. how many nematodes was on that plant, with the genotype of that plant, so which genes did it have. And in so doing, we can work out which parts of the Arabidopsis genome contribute to that phenotype. And so there you can just see the graph of or the difference of all the different lines. So this will be the most susceptible and the most resistant. And then we can correlate the phenotype with the genotype to highlight parts of the Arabidopsis genome that contribute to that. So in this case, basically it's the black dots. So there's some black dots on chromosome 1, some on 3, and some on 5 that show you where the genes are in Arabidopsis that contribute to this phenotype, i.e. a resistance gene or something like that. Now we don't just have one phenotype. I listed several phenotypes, right? And so we can go through this process for all of them, and then in so doing, we can annotate the entire Arabidopsis genome for the nematode resistance traits or nematode traits. And so I won't go through the detail, but you know, there's a lot of stuff, right? So basically, we've gone from 0 understanding of which genes in Arabidopsis contribute to these traits to all of this in like a year, and that's including the time to make the machines, to design and print machines. So I think it's amazing what you can do with a 3D printer and AI basically these days. Okay, so yeah, that's basically where I want to end. What we're doing now is kind of the obvious stuff, so you know, looking into the dynamic phenotypes and things like that. I think for me, the dynamic phenotypes are the most interesting because, you know, for most of plant breeding history, when you're thinking about pathogens, what you look for is the the smallest number, like less is good and more is bad, right? But this kind of opens up your way of thinking if you start thinking about dynamics, because you know you're going to harvest at day 90. So growth rate, for example, might be really interesting. So maybe you don't care how many nematodes infect the plant, as long as they grow so slowly that by the time you harvest, they haven't had a chance to complete their life cycle. The following year then, your field is nematode free. And this is like the holy grail, because once these nematodes get in your field, they're there forever. And so this kind of, you know, thinking about the dynamics of pathology, not just the black and white, has really changed my thinking about crop protection solutions. Okay, the last thing I want to do is just thank everybody, and I literally mean everybody. So everybody in the lab had to go at some point to the greenhouse to image plants. And so everybody helped, and so I thank everybody for that. Most importantly probably is the AI team, because without them we'd probably still be counting lemons. And then of course the funders and you for your attention.